Welcome, grade 12. Welcome to our series of lessons that are brought to us by VT Africa Holdings and its subsidiaries, Fibotech and Fastratech. Is always my request. I request you to log on to www.smart.com to take part in the assessments, and I would request you to use the ID 598751, and then you add your name, your real name. Once again, our login details, www.hellosmart.com. Sign in as a guest using ID number 598715. Thank you very much. Sorry. Right. Sure you have logged on. Thank you for that. Yesterday, we end up our lesson wrapping up on Mendel's laws right. We ended up looking at, we looked at three Mendel's laws of heredity. The law of dominance, the law of segregation, separation of segregation, then the law of independent assortment. Right. So as a wrap up of what we did yesterday, let's quickly try out the following questions. Very quickly. Right. It says, what's the name of the geneticist who would first describe heredity laws? And our answer is obviously Mendel. I will wait for you guys to log on. Okay, let's do it together. Some network problems are affecting, they are persisting and logging on. Let's move on to the next question. Question 7. Mendel's law of heredity includes the following. A, law of segregation, law of dominance, law of independent assortment. All of the above, yeah? Without wasting much of time. All of the three laws relate to the laws of Mendel. Segregation, sorry. Law of segregation, law of dominance, not co-dominance. So B is wrong. And then the law of independent assortment. Okay, so I do have answer A and C is my answers. I had read codominance as dominance. Next question. My network is not as good, so it's noted there. Okay. Which law states that, because I'm having problems here, let me go back to my which law states that the alleles separate randomly during gametogenesis? This is part of our phase of meiosis, as I challenged yesterday to go and check. So which law states that alleles separate randomly during gametogenesis? The law is covered under independent assortment. Under independent assortment, there is no formula for the lining up of chromosomes in metaphase 1 of meiosis. So the best answer is the law of independent assortment. Next question. Or which law states that recessive genes only be expressed in a zygote? The recessive genes are only expressed in a zygote under which law? Only when there is dominance. So the answer becomes C, the law of dominance. Next question. Right. It takes us to Punnett squares, but I would like to use a different approach to Punnett squares, because Punnett squares is only a part of what we use when we are solving our genetic diagrams. So what I want us to do is we continue to look at a scenario of codominance. All right. After codominance, we look at incomplete or partial dominance. Then we look at the genetic diagrams. Okay, that's where our Punnett squares do come in as a way of helping us to solve some genetic diagrams. Remember, we use our genetic diagrams to solve problems whilst we are looking at one trait or we're looking at two, giving us either monohybrid diagrams, or if we're looking at two, then there'll be dihybrid. So that's what we are looking at. But today, I want us to discuss, on top of what we've discussed, codominance. After that, incomplete dominance. Then we now look at how to solve genetic problems of monohybrid nature, and then of dihybrid nature. on the lessons that are coming, right? So when we say co-dominance, in a scenario where we have co-dominance, co, it means there is a single trait. However, there is a single trait with two variations. Yesterday, we looked, or the day before yesterday, I gave you an example of being able to roll your tongue and not being able to roll your tongue. We also gave an example of being able to bend your thumb and not able to bend your thumb. So if we are looking at the trait of thumb bending or not, or tongue roller or non-tongue roller, that's the trait. Then the two variations will be you can roll your tongue or you can't. I just want you to stay abreast with the terms that we use. Okay. However, neither variation is dominant over other, but the two variations are expressed in the phenotype. If you remember, when we're talking about the laws, the very first experiment that Mendel was doing, when he bred white colored peas, he got the offsprings that were white colored, which means that was pure breeding, I mentioned yesterday, and it was complete dominance. Sometimes complete dominance is also referred to as symbol dominance. Grade 12, you should now learn to master the terms that we use because they will help us to understand some genetic questions that we'll be solving. All right. So rather than in the previous example of all white from white offspring, symbol dominance, we have a scenario where the phenotype is expressed. Okay, my first example here is in humans, where blood groups in humans, variations in blood groups, we have got A, B, and C as our blood group. Neither A or B is dominant over the other. What do they mean? It means that for one parent, as an example, if one parent is blood group A, then the other parent, mother, is blood group B, for an example, the offspring cannot be blood group A only, but the offspring can either be blood group A, B, meaning neither A nor B is dominant over the other. So there is co. If you are co-directors, if you are co-managers, Mr. Manager Tato and Mrs. Manager Ntabi saying none of the two is more superior or has more managerial powers than the other. They share the duties responsibly. That will be the scenario. All right. Another example of co-dominance is the fair color in cows. The fair color in cows is the trait. That's trait 1. Then the variations can be either red or white. So in this case, this fair color is not dominant over each other. So red and white. The alleles for both red and white color produces red and white hairs. So you find red and white hairs on the color. And we can say it's a rowing color, which means neither red nor white is dominant over, but they are co-sharing their color. You can see red flares, you can see white flares on the individual, on the animal. That is codominance. You would consolidate this when we come to our problems that we can find in our genetic problems. All right. I'm sure you're following. Now let's look at incomplete or partial dominance. Incomplete or partial dominance, neither allele show dominance. Don't confuse it with core. In this one, neither is dominant. Okay. Neither is dominant. However, they meet in between. They blend. An intermediate variation is therefore produced when a condition is homozygous. That's very important to note. When the condition is homozygous, which means when the condition in the offspring is not homozygous, we will get a different outcome. But for now, let's take it stage by stage. In incomplete dominance, the offspring is intermediate when the genetic makeup is heterozygous. Remember, I emphasized you don't need to claim these words. You know what is a heterozygous, a hybrid. Both alleles are present. We get heterozygous, which means in this scenario, to just take you back, if it is a simple dominance or a complete dominance, the heterozygous condition will show the dominant trait. That's using T and T for my letters for the example that I'm giving you. Remember, we said when we're looking at Mendel's codominance, T being a dominant trait and T being the recessive. If it's a complete dominant, the phenotype of this would resemble T because it's dominant. But in this instance, we will not have T being dominant over T. Rather, T and small t will both express the effects of their genes will be expressed on the offspring. So it will be a intermediate color that is in between. All right, let's look at the example. Flower color in the snapdragons. Snapdragons, it's a kind of a fly. So our trait here is the color of the flower. The contrasting forms are red and white. Red and white. When they meet, their offspring is pink. So the blend between red and white, they did not give us either red in a dominant setup, if red was dominant over white, or vice versa. If white was dominant over red, we don't get a white in our heterozygous. Rather, we get a blend. So the effect of red and white came out pink. That's partial dominance or incomplete dominance. Can you see the difference from this example alone? I know it would make sense as we solve other examples. With codominance. In a codominant setup, as an example for this, hair color in cows, white, red and white color produces red and white hairs, which you can give the color. Sorry, the color can be called roan, but you can really see that there's red and white. I'll give you some diagrams for similar colors on the next lesson when we're doing our revision. You see it comes out as red and white, right? Both are there, cool. But when you come to partial, we have pink. Pink has been blended as a result of red and white, right? Okay. Today we are doing an exercise together. Class, I want us all right. Let's solve this problem. For each question, I want you to identify for me identify the traits, the variations of the traits, and the type of the genetic problem. The traits, the variations, and the genetic problem, right? When a red cow is crossed with a white bull, all the calves produced are roan. Remember, this question is summarizing complete dominance, codominance, and incomplete dominance. Okay, when a red cow is crossed with a white the cows produced a roan. And roan is a mixture. Roan is a mixture of white and red. So in this case, both white and red is an effect on the outcome, on the offspring. So grade 12s, what is the trait here? What is the variations and the problem? Right. So the trait here is fair color, the color of the animal is our trait. And the variations, red and white. Right. And obviously, it's a codominant problem. It's a codominant problem. The genetic problem is codominant. Very simple. So the understanding the question will help us not to cram the definitions, will help us to solve genetic problems. Right. Oh, sorry. Let's look at question number 2. In garden peas, tall is dominant over dwarf. In garden peas, tall is dominant over dwarf. Condition, scenario. If a homozygous dwarf plant is crossed with homozygous tall plant, all plants are tall. Good. Very straightforward, simple question. All right. Oh, I'm fine. If homozygous dwarf is crossed with homozygous tall, outcome, all plants are tall. Very simple question. Identify the trait, the variation, and the type of the problem. Trait is height. Trait is height, grade 12, either tall or dwarf. That's the trait. So it's the height. All right. Variation, variety, tall or dwarf. All right. And what kind of a problem is it? All plants are tall. Homozygous, dwarf, homozygous, tall. All plants are tall. So it's a symbol. It's a symbol or complete dominance problem. Okay, you see, I'm a bit slow today. Some feedback from my previous lesson is saying, Sydney, at times we're a bit too fast. Okay, so I slowed a bit today so that we can understand our chapter. We need to get all the 45 marks of our paper 2 on inheritance and genetics, right? I'm a bit slow also today because I'm introducing you to solving genetic problems, and terminology is important. Understanding the hints within the question is important, right? In this number 2, it's not always that you'll be told that homozygous plants, dwarf plants, or homozygous tall plants. No, I can remove the term homozygous in this question, but you still be able to get the answer. I can simply say in garden P, tall is dominant over dwarf. I can tell you that. And then I said, if a tall plant, if a dwarf plant, I eliminate, is I can just delete homozygous here to still make sense. In a garden piece, tall is dominant over dwarf. If a dwarf plant is crossed with a tall plant, all plants produced are tall. From this information, I'll still get what I need to get. All trait, height, variation, tall and dwarf. Problem, I can still deduce that it's a. I can still deduce that it's a symbol or complete dominant problem. Making sense? Hopefully. So, thank you very much. So we have covered complete, co, and incomplete forms of dominance. And we're looking at just one trait at a time for each problem. Okay. Okay. Today we started with an assessment. We did an assessment at the middle of our class. Let's continue to where I want us to get to. Right. Let's look at how to solve genetic problems. Examples we're looking at just now are examples of genetic problems. Right. We use genetic diagrams to solve genetic problems. Grade 12, are we following? Right. Some of the things I said it on the first lesson, right? A genetic diagram shows characteristics, how characteristics are passed from one parent to the offspring. Remember, we said genetics and heredity. We are studying how information is passed from parent to parent. All these terms are there on my vocabulary on the first slide of our first lesson. Okay. In a genetic diagram, the following symbols are used. Remember, we said geneticists, they use alphabetical symbols to represent alleles. We also use symbols to denote our genetic diagram. Right. In a genetic question, if I see P1, Sorry. P1, I know it represents the parental generation. The mother, the father, the paternity side, and the maternity side. We represent them using P1. F1 represents first generation. If Sydney met with his wife and produced a cutie baby girl, that's a first generation of my process of passing on my genes from parent to parent. She grows up, she learns genetics, she's an entrepreneur, gets married, she become P2 of my generation. So she becomes F1 of the generation of mine, which means she has grown up to produce my great grandparent. So it becomes a second generation denoted by F2. You see, it's important to know these symbols because when we are giving genetic questions, we normally refer to them. It saves time and confusion when you are solving problems. Right. Whatever I said here, I've said letters of the alphabet represent alleles of a gene. We mentioned it yesterday, and we gave an example. Right. Very poor drawing from myself. Okay, I wanted to come closer home. These are my chromosomes, joined by a centromere. Okay, certain gene occupies this space. So this gene, maybe it's for coat color. When I'm representing coat color from my previous example, it could be either red in a blue or it could be white. So the alleles will represent this allele and this allele. Alleles of a gene. That's where it comes from. The same letter is used to represent both the dominant and the recessive allele because we are referring to one gene. We don't say X and W, which I say don't use letters that confuse the upper and the lower case. All right. The first letter is the dominant form. It's a repetition. We mentioned this, but we need to emphasize it. Of a trait is chosen as the symbol for the gene. Okay. Right. With the example showing genetic diagram. How do we go about it? Okay. It does not clog our working space. Right. In garden peas, toe is dominant over dwarf. If a homozygous dwarf plant is crossed with a homozygous toe plant, what will be the appearance of the F1 plant? What will be the appearance of the F2 plant? Okay, for today, Let us ignore the part 2. Just ignore part 2. So there's my question. Homozygous dominant. TOR is dominant over dwarf. If a homozygous dwarf plant is crossed with a homozygous TOR plant, what will be the appearance of the F1 generation? What will be the appearance of F1 plants? It's difficult to just tell me, no, the appearance will be TOR or dwarf. You need to do a genetic diagram. So copy this question. We'll start with it on the next lesson. But importantly, I want you to follow the steps. Solution: first, define your genetic symbols. So let me help you. Which symbols would you want us to represent our traits? Since it's height, TOR, and dwarf, let me take T. So T will be the symbol. Second, state the parental phenotype. So genetic diagram should follow this. Parental phenotype, TOR, and dwarf. Third, state the parental genotype. Parental genotype. What is it? For TOR, it's T and T. It's a genotype for a parent, so it has to be showing all the alleles. For the other one, dwarf, homozygous, T. Right. Fourth, show meiosis. Gamete formation. Show the gametes. Show random fertilization. And I've said here, we can use a Punnett square. Some of my learners always think that a Punnett square shows all these stages. No, a Punnett square only shows part of the problems, random fertilization. Which gamete of this one can meet that one? That probability, that chance of this and this meeting is the only thing that is shown by the Punnett square. So when I go to the Punnett square on the next problem, sorry, on the next slides, on the next lesson, please, we are continuing from here. We are only showing that probable chances of genes meeting. Then we show the F1 generation genotype. We show the F1 phenotype. We show the ratio. It's important to know the ratio. Why? It will quickly help us to solve problems because there's some things that they did not. Okay, so follow this for your homework to solve this problem. Send me messages if you are having problems. Thank you very much, great girls. We meet on Monday at 2 o'clock. Enjoy your day.